All right, welcome everybody. Thanks for joining for our weekly webinar on COVID-19. I'm grateful again because Bernadita Mendez and Mary Gutierrez are both joining us as panelists. So thank you guys for joining and being with us. It's really, really appreciate that. So today we're going to talk, I'll give you an update a bit about the statistics of where things are. And then we'll talk a bit after that. We'll talk a bit about diagnostic testing, both a bit about the PCR testing and then a fair amount about antibody testing. So since that's really become a big topic as we think about, okay, how are we going to get people back to work? How do I know if I'm immune? Do I need to still be worried? Things like that. And then we'll talk a bit about treatments at a high level. And then we'll talk a bit about mental health and spend a little bit of time talking about sort of how things are feeling amongst ourselves, and also talk nationally what people are talking about as well. So that's sort of the overview. Goal is to keep it under an hour, and hopefully we'll sort of talk for the first 30 to 40 minutes, and then we can do Q&A. So if you do have questions as we come along related to what we're talking about, or there might be a topic we're not covering, please feel free to put something in the Q&A button. I'm going to launch a poll right now that just asks what topics you want to spend time talking about, and that way you guys can weigh in on what you feel most passionate about, and you can choose more than one option. So I listed here PCR testing, antibody testing, treatments, conventional treatments, herbal natural treatments, statistics, or maybe there's some other topics. And if there are other topics that I didn't list, then of course feel free to just throw them in the Q&A, and that'll allow us to get a good view of what people want. Okay, so we've got a lot of people, pretty much everyone wants to talk about antibody testing. Good, that's what I want to talk about today too. And then a lot of people want to hear more about conventional treatment. So we're going to talk a bit about that. And then there's a fair amount, 40% want to hear about natural and herbal treatments. And I don't have those in my slides. We can talk about it. If you want to serve up a question, feel free to. Last week, I think it was, in my previous talk, we spent a fair amount of time talking about herbal and natural supplement treatment options. And there's a range of slides there and some conversations. So if you want to go to the YouTube channel and find my talk from last week, you can. If you have a specific question, feel free to throw it in. We'll cover some stats too. 30% of the people wanted to do that. So, and then 40% about PCR testing. So great. So thanks for that. So let me go ahead and just launch these slides here so you guys can see them. Let me share my screen. Great. So I'll just give you an update. Some of you probably have seen this from other places, and this is from the Johns Hopkins Center. So we're now up to 2 million confirmed cases. So it's basically doubled since last week, and total deaths at 133,000. And you can see here, United States is ranking pretty high, with New York, amongst all countries, New York ranking 5th as one of our states. So I think it might be interesting to look at sort of over time how things have changed. And so I showed this last time. This comes from the Financial Times, where they pull in a whole bunch of different data sets. And this looks like the daily deaths with a 7-day rolling average. And you can see here that the UK and US, their death rate is still trending upward. It's slightly shifted, you can see, whereas you can see Italy and Spain is actually starting to trend down. And so you can see sort of the arc here. Hopefully in a couple weeks from now, we'll start seeing our UK and US trending downward, but if we have some time to talk about how we re-enter into socializing again, as you've probably heard, there's going to be some serious conversation about how we do that right so we don't end up with more cases. This is just a different view of that conversation, and I liked it because it shows where the impact is greatest. And you can see here over time, this is looking at the proportion of deaths at the bottom. So it doesn't really look at the number of deaths per se at the bottom going across there. It looks at the proportion. And you can see in the beginning it was Asia, and you can see Europe in the blue, the Middle East in green, US and North America growing. And now you can see where we are now. We are comprising, as a United States in particular, it says North America, but the United States in particular is comprising a large proportion of the total deaths in the globe. The upper graph, you can see a different version of that. So the US now accounts for more than 1/3 of global daily deaths. And of April 15th, 2,408 people. And the total deaths were 6,891. You can see daily deaths. So the ship, you can see how it's moving really across the globe. And we're hoping that, remember, deaths are sort of there's a huge delay between cases and deaths. You know, it tends to be like a 3-week delay at least. So we'll see how our caseload changes. And then later the death rate will follow that. If we come in just to the United States itself, you can see here by color coordination where the predominance is. And you can see how great California has done relative to others. This is adjusted for the population density or population overall, and the darker colors being the more affected places. So up in the Northeast, unfortunately, quite significant. And I believe that's Louisiana in the South as well. And Mary's in Florida, and that's not doing too bad, but we'll see how that evolves over the next month. And you can see Hawaii is doing quite well. In the Bay Area itself, you can see here, Santa Clara, last time I showed this was, I think, 2 or 3 weeks ago. They had 30 deaths. They were leading. They were the #1 county in all of California. And I believe they still are, and they're at 65 deaths in Santa Clara County, just south of us here. San Francisco, you can see here, and then Alameda. And so you can sort of see the cases and then the total deaths. San Francisco's done, if you remember, really early on, they did an early lockdown, and that's really, thankfully, caused a huge positive impact, as well as the surrounding counties in the Bay Area. So that's an update on the testing. I'm sorry, on the statistics. And now just to talk a bit more about diagnostics. So again, just for review, there's oftentimes people aren't super clear about this. And I just want to really make sure that we're clear to say that there's 2 different types of diagnostics we're thinking about. There's the PCR testing, which is the acute test to see if you're acutely infected. Do you have the virus? It tests for the actual virus. It looks for the virus in your body. And we do that typically by taking a swab that has a brush on the end of it, basically, and a long stick attached to it. And we put it through the nose into the back of the throat. And then it's called nasopharyngeal swab. And we sample it usually for about 15 seconds and then pull it back out, put it into a medium, break off the tip so it fits in there, screw it on, send it off to a lab. It's a very technically challenging procedure, and also it's very dangerous because you have live virus sitting in this container, and the lab people that are running these tests have to be extremely careful handling this specimen. It's prone to air. It can be very good in a highly trained facility, but it can vary from place to place on how good that facility is. So you may have heard some people have been negative, yet they're pretty convinced they had symptoms. And one of the studies I'll show, there was a subset of people that were negative, but they had other findings, like on CT scan and a clinical presentation while they were hospitalized, that the doctors were pretty convinced they had it and the test was a false negative. And sure enough, they developed antibodies later, and we'll talk about that in a moment. The second category of testing is antibody testing. And what is an antibody? It's a protein that is a part of our immune system, and it says, oh, here's a foreign entity. I need to develop a military against that to fight it off next time it comes in or while it's here. And for in this particular case, there's antibodies that we developed for the SARS coronavirus 2, so COVID-19. So when we think about antibodies, we can think about total antibodies, and then specifically two big categories: IgM antibodies, which is usually the acute phase reactants, the first sort of foot soldiers to come to the event. And then you have IgG, which comes in later. And they come in after the IgM, and they usually show up and have ideally a sustained antibody presence. So that ideally, doesn't happen in all cases. For example, the common cold, it doesn't happen. But in other conditions, it may, where you have antibodies to something and then you won't get reinfected again. We don't yet know what that looks like for COVID-19. So thinking about that, there's a lot of different companies out there. I'm being approached by a lot of different reps that want to sell their antibody testing. The FDA has recently changed their rulings and now requiring all tests to go through and be the Emergency Utilization Act, and so go through that pathway. Now a lot of these distributors are changing their who they sell to, and are now only selling to pharmacists, or they themselves are setting up shops and you're going to directly run the testing themselves. So there's a range of there's a very fluid marketplace and very fluid with the regulations. And I've gone back and forth, and we'll talk about this with the panelists, with Bernadita and Mary, that I've gone back and forth that should we start using these tests or not? What are the pros and cons? And we'll talk about that. So if you think about the different types of places you can go to get tested, for the PCR testing, I want to the new thing to add to this slide is Project Baseline COVID-19 is called, which is by Verily, which is one of the alphabet companies, which used to be Google. Google then renamed itself, its parent company called Alphabet, and there's Google that's separate. I think it's still a subsidiary. Google is of Alphabet, although that may no longer be the case. But Verily is one of these companies, these alphabet companies, and they're looking at human aging and looking really at the biosciences. And they focus now on COVID-19. And I will say that before I move from this slide, that there is improvement by Quest and LabCorp on their throughput. They've actually increased their ability to actually run these tests now. If you remember, there were 2-week delays on them, and it seems like that's clearing now. Of course, in the Bay Area, is less of a need. There's fewer people that at least in my experience, in my population of patients, as well as people around me that I know of, very few people are becoming acutely infected. But there is now more ramp up, more capacity, and therefore their delays have now been much improved. So here's the website, just to let you know about the COVID-19 testing program. They've opened up a site now in San Francisco, which is new. It wasn't there last week, and now it is. So they're popping up sites everywhere and growing over time. And they've been doing a long-term community-based cohort study in general, and now they've added this COVID-19 piece where they're actually doing testing. So you can click here, fill out some screening, and then identify if you're eligible to be tested. So worth knowing, it's free, and it's part of their research though. So they have a back-end research protocol that they're running this through, and it was developed in collaboration with the Department of Public Health for California as well. So that's something to know about and let other people know about as well. All right, so let's chat a little bit about antibody testing. So if you remember, we talked a bit about sensitivity and specificity, which we'll talk about in a second. But basically the bottom line, the great thing about this thing is you're not dealing with a live virus, right? You're dealing with blood. So you take a little finger stick and then you draw this blood, drop it onto this little cartridge, and then you get results on the little cartridge, anywhere from 2 to 15 minutes. You get these results. So what is the result showing you? It'll show you whether you have nothing, negative, or you have IgM, or you have IgG, and which is great because it's so quick, it's easy to use, it's pretty foolproof. It doesn't have to go to a laboratory. You do it right there, done by a clinician, typically a nurse or medical assistant, can do this and process the test. And within 15 minutes, while you're there, you know your result. Really simple, easily scalable once you have a good test. And you can imagine doing this at work sites. You can imagine just doing this at the pharmacy. This can be done quite easily. This particular product is from South Korea, called BD Biosensor, and their specs show, this was a relatively small sample size, but they showed 88.7% sensitivity. So that means that only roughly 11% would be false negatives. So meaning that you might have antibodies, 11%, 100 people were tested, 11 of them would have a negative result, even though they actually had the disease. And then the specificity was pretty good as well. So just to review for a moment, sensitivity and specificity, we did this last week. But the sensitivity is when you think about, you actually know you have the disease, you go and take the test, and does the test give you a positive result or not? And if it's 90% sensitive, that means 1 out of 10 times, or 10% of the time, it's going to give you, it's going to be falsely negative. So the result will be negative when actually you have the disease. Specificity is the opposite, meaning that you don't have it, and but you're being told you do. That's the false positive one, which is in the upper right-hand corner, the FP. So back to sensitivity. Sensitivity is really good if you get a whole population of people and you want to test them all and you want to catch everybody. It's like a big net you're casting into the ocean, and you want to get all the fish, and then you discard the ones that you actually don't want. Hopefully you're keeping those fish alive. But that's sort of what you're trying to do with sensitivity. You're trying to catch everybody, and then you go and find the ones that really do have the problem. What I mean by that is a lot of times you give up specificity when you try and increase your sensitivity. And so what happens is you end up, with a test that's highly sensitive, you'll get everyone that's truly positive. And then that's great because you won't have any false negatives. But then you need to confirm that everyone is truly actually has it, that you don't have any false positives. And for that, you do a different sort of test typically that will have that does a really good job of ruling out, making sure that there are no false positives. So here's a study where they did this sort of testing. It's a complicated slide, but I'll just hone in on the important parts. The big important part is 173 people. So that's a reasonable number of people. These were folks who were in China. These folks were all hospitalized, and they looked at them by their severity of their illness and a range of other things. They followed them for about 30 to 40 days. And what they did here is they looked at two different tests, the ones we've covered already. The RNA test, which is the PCR testing for the virus, and then the antibody test. And the antibody test, they were able to calculate the total antibody, which is the IgM and the IgG, if either one of those were positive, as well as the separate ones, the IgM and the IgG. And then there's a combination of all the above. So they basically took on the far right-hand column is the PCR test and the antibody test. And were you positive for any of those? So if you just look at the the virus testing, which is the RNA testing, and this is all sensitivity, right? Meaning, how good was this test at giving you a positive result if you had the disease? So if you look here, the total was 67%. So it's not super good at catching everybody. Now, why is that? It could be problems with the swabs. It could be problems with the technicians doing the actual test. It could be problems in the labs. But what's very interesting here to note is the first 7 days, onset of when they presented, only 2/3 of the people ended up with a positive PCR test. And then, of course, over time, as your virus, wanes, because by week 2 and 3, the virus, your body starts to clear the virus, it becomes more and more difficult to see the PCR test. You see the number goes down. It goes down to 54.0, 45, and then 45.5. So only half the time was there virus found in the person. But at its best performance, it was 66.7%. So not fantastic. Now, if you look at days 1 through 7, and then you look at the antibody testing, it also doesn't do great. If you look at the next column over, it says 38.3. That means only 38% of the time that the antibodies show any positivity in the first week, which makes sense because it takes a while for the body to mount that immune response. But what happens by week 2? Then it goes up to 89.6%. And by week 4, so day 39, if you will, right? 15 to 39, the bottom row, it says 100.0, I mean, everybody was antibody positive. And then they broke it out. What do you mean by antibody positive? So if you go over, you see IgM and you see IgG. The point of this slide is the combination does really well. So if you look at the far right column, you can see there that 78.7%. So days 1 through 7, by using the combination, you get 78.7%. So if you do the PCR test and an antibody test in the first week of showing up and you think you're sick, it's much better than just doing the PCR test by itself, which is currently what everyone's doing right now, just the PCR test by itself. And then as you move into week 2 and 3 and 4, you can see on the far right-hand column, it says 97.0 and it says 100%. So again, you improve your test characteristics, particularly day 8 through 14, that time zone, you can see it's better than the total antibody, which is 89.6%. By doing the PCR testing and the antibody testing, you move from 89.6 up to 97%. So the combination really seems to be a lot better, which we're currently not doing. So this is it's only one study. We'll have to look at this, this is their test that they were using, their ELISA test. And it's only 173 people, but pretty interesting. So I thought I'd take just a moment. I'd love to get some input from the panelists to see if you guys have any thoughts and comments about some of this testing.
Hi, Brad. Hello, everyone. I think that's a great overview, Brad. And before we hopped on the call with everyone, we were having a small discussion with Bernadita about the ability to roll these tests out with a great deal of confidence. And I, for one, I know everybody is very eager to see this happen as quickly as possible for a lot of different reasons. But as we agreed in our little talk, that time is needed to ensure that these tests are truly effective. We don't want to roll something out that is actually going to skew the reality of the situation. And it takes time. For those of us who have worked in labs for a while, we know we go through a period of troubleshooting. It's trial and error. You start tweaking the assay results. You're comparing results to controls and to numbers of people you have examined or you've included in your studies. So there is a bit of time, and Brad had inquired earlier as to at least my opinion on this. And I feel that we need to be looking closely at all these fabulous studies that are underway and make an informed decision as to which ones have sufficiently validated, as I call it, validated the assay for use. And I think the FDA, obviously, and others recognize that there is a process that we must follow, which is why they have designated this as emergency use only. But we're making great strides. I mean, I think this is excellent work moving forward. It's just going to take a little bit of time to have proof of concept. I don't know, Bernadita, did you want to comment?
Yeah, I agree. I agree with that. But I also think that the fact that if there is the possibility of using the tests in combo, as Brad was pointing out, that is, you can use the reverse transcriptase assay to look at the virus itself, and at the same time do the antibody assay. Your percent of certainty increases, even though each one of those assays is not perfect. So I think because of the situation we're in today, I understand that probably that's what FDA is trying to do. Put minimum standards in terms of, specificity and sensitivity. Maybe they're not going to be 100% or 99%, which is usually, when you're going to blood banks or something like that, you shoot for very high. Probably they're going to accept things that are not in the very high range, and we're going to see like generations of this test evolving, getting better. But at the same time, if we're able to do them in combo, we're going to increase that certainty because we have data from a couple of different assays. And eventually, we're probably going to develop a third kind of assay that Brad mentioned on passing, which are called confirmatory assays. So, you get somebody who has antibodies, and because we develop an assay that maybe the sensitivity is high, but we're going to get some false positives, well, we don't want to call positive somebody and send them back to work if we're not 100% sure that they have the antibody. Then we'll have a backup test that person will have to have their blood sent again to do an extra test to make sure, a test that's going to be much more specific now, that's going to make sure that that person has the antibody. So I think, we're in the midst of it. We have to be careful, but the only way to get it done is to start using them. I think that FDA is going to hopefully provide a first level playing field and check across the board.
Yeah, that's an excellent point. Sorry, Brad, go ahead.
Okay.
Oh no, I was just agreeing with that one point, making that combination assay or test regimen, if you will, is an excellent step in the right direction. One thing I also wanted to add is that one thing that I've been considering, and I'm not sure what the case is with this, I would imagine there's a lot of people who are starting to use these assays for the first time because there's been such a high demand for performing the tests. And so there's going to be a little bit of a period of time of people actually learning how to do this right and interpreting results correctly. It won't take long, but because the assays are, as Brad pointed out, not that challenging. I would say, from my experience, the serological assay that Brad mentioned, which I call a rapid diagnostic test, an RDT, that's far easier to perform than PCR. PCR is a bit challenging. It's very easy to do PCR, but it's also easy to have problems with PCR for various reasons that I won't go into here. But I think, as Bernadetta, you said, that combination is excellent step in the right direction. And if we can have confirmatory testing, and another thing that I don't know whether or not they're doing is performing these tests in duplicate or triplicate. That's also an important feature, and I'm not sure if that's happening quite yet when we're talking about such limited testing.
Yeah, so there's a few comments on that. So I know that there's studies going on now. There's one that I'm hoping to help get funded that's going to do sequential seroprevalence testing. So there's two approaches, right? You can do it in real time, so you're positive or negative, depending on the test characteristics, and you repeat the test, like you're saying, and duplicate. Should it be done by another? Should it be, if you're false positive, are you reacting to the reagent itself, or could you be cross-reacting to another coronavirus? And that's what the antibody test is picking up, is you had a different coronavirus, and that's why it's giving you a positive result? Or is it, because the reagent that you're reacting to? or I presume there's other reasons. So to repeat it either from the same company or a different company. So that's one possibility. The other, in this case, for this study, for example, is going to do it sequentially over time. So at a population level, they'll look at herd immunity. And also, to your point, it would say, if it's positive, is it positive again a month later? That looks at a different conversation, which is how long do our antibodies last? Which is also the other question. How long, are we immune if we have antibodies? We don't know if that means we're immune or not. Apparently with dengue fever, I believe that means you do worse if you have antibodies to dengue. If you get infected a second time, my understanding is you actually have a worse reaction to dengue on the second infection, right? Yeah. So if that's the case with this, well, that bodes negatively. Well, so, which we don't think, I believe we don't think is the case, but worth mentioning. And then I guess the other question being posed in the audience is, what can people do differently? Let's say you get a positive test for antibodies this week. There's a group up in Richmond right now that's doing it. They're charging $120 per test, which is enormous if you ask me, because I know the true cost of those tests. And they say, what's the point other than, is it just curiosity is the question, or what can I do with that information? I have my thoughts, but I'd welcome what you guys think.
Oh, for me, are you asking?
Yeah,
Hannah?
I'm just curious if you guys have an opinion.
Sure.
You can say you don't have one, that's fine, because it's.
Yeah, no, I am a scientist at the core, so I want to be really thorough about any results I get and I interpret. And so if someone is antibody positive, I want to understand at what course in their in the process of infection are they at? Where are they at in that progression of potential disease, or whether it's asymptomatic or symptomatic? Somebody might have a test that's positive and they're at the early stages of infection. Not that early, because as you pointed out, there needs some time for the system to respond to the virus in their system. So for me to sit there and say, okay, I have a positive antibody result, what does that actually mean? We need to understand what those results represent in order to then elicit steps that one could take. Can one now go outside and go back to work and go to the stores and such? Or does this mean that you're at a different stage of disease that may preclude you from doing that? That's just my very quick off the top of my head answer. Bernadita?
Yeah, I think that the million-dollar question is, in fact, whether you're protected if you have antibodies, isn't it? I mean,
absolutely.
And certainly we don't know that. And part of the reason we don't know that is because we haven't done enough testing. Right. Because I think if we went out there and found that a big chunk of the population doesn't have any symptoms, and they have antibodies and they've been going out and about, it's likely that we could hypothesize that they are protected. What we don't know is the length of the protection, and for that we would need to do like long-term follow-up of the antibody. But before getting any information, we're still a little in the dark. So it's hard to make decisions when we're talking about, the workforce, people going back to the workforce. We're assuming, and I think that, there are a lot of people that are much more experts than what I am in terms of extrapolating from other coronaviruses, and what we have seen in terms of immunity with other coronaviruses that can extrapolate and say, well, maybe if you have IgG, you're going to be immune. And that's the reason why we're interested in doing this testing today, to start deciding whether there are people that can, be a little bit more mobile and be out there again, people that are not in the high risk groups and so forth and so on. I think it's very valuable information to start generating. I'm not sure that we have 100% the responses today, I think it's very useful also to produce, to get antibodies from people that have those antibodies, to use them as therapy for people that are sick, which is another part of why this thing is useful, why the antibody test is useful. But I'm not sure that we can be 100% certain of how we can use the test today.
And that's a very good point that you just made, because I was wanting to jump in earlier and talk about neutralizing antibody. Mm-hmm. And those tests need to be conducted. There are certain viral infections that react very well to what we call immunotherapy. So delivery of antibodies, you might hear of monoclonal antibody mixtures. In fact, when we were vaccinating against smallpox, we were using an immunoglobulin mixture against another type of pox virus. So I'm very interested, having worked in immunotherapeutics, as to whether or not neutralizing antibodies that will do exactly that, they will neutralize the virus's ability to attack, if that is possible with coronavirus 2. Mm-hmm. So I know they're doing that right now, and
yeah, they're doing.
Yep. Yeah. So the study I was referring to that is out of UC Berkeley, actually, they're looking at neutralizing antibodies as well. So the plan is to take blood on a subset and look at neutralizing antibodies, compare those results with the antibody on the finger stick results as well. Look at just those and look at those characteristics across all that. Right. So I think the question really relates to, I don't have symptoms in the past 2 weeks. I want to go back to work. I had symptoms a month or two ago, or maybe I didn't. Give me a test, if I have a positive antibody, I'm in the clear. It means I'm immune, right? I can go back to work. So that's the question that we don't know the answer to yet. We think it's probably true, but I think the first data will come out from healthcare workers, because we're the ones that are going to get sick. 19% of the recent MMWR showed that, I think it was 19% of those that they studied were healthcare workers. Of the cases were healthcare workers, because we're 5 times more likely to get it because we're in the thick of it, the front lines. And so we get infected and we get better, and then we go back to work. Right now we're immune. Yes, we're antibody positive. I want to go back in. That's what a lot of us want to do. And so we'll find out because we'll study us and know in the next turn. I think the odds are pretty good because we can look at China and Korea and see that. I think that there's a good chance of some immunity there. I think otherwise we would have seen a larger resurgence, I think. But for how long? Does it last 6 months, a year? That part we don't know. Okay, well, great. Thanks for those thoughts. I think hopefully that's helpful to everybody. So let me go back to our slides and talk about treatments. And we already alluded to one of them. So the Milken Institute's been doing, they're a great source to look at sort of the pipeline of treatments that are going on. And they really have broken it down like this. So you can see there's 79 vaccines being looked at as we speak. It's a lot. I mean, just remember how long this has been around. Not very long. I mean, this is an amazing amount of clinical research and laboratory and clinical research and translational research that's happening as we speak. And then on the treatment side, you've got antibodies, antivirals, and then a whole big chunk that's considered others. And then you have cell-based therapies as well. And then you have some RNA-based therapies. And then scanning compounds to repurpose. So existing treatments that are out there for other things that they're trying to repurpose for this particular situation. And you can imagine hydroxychloroquine, for example, as Plaquenil would be sort of something like that. Here's sort of, there are 50 treatment candidates that are currently in clinical trials as we speak, or being used sort of under quote compassionate use. Or like remdesivir is sort of in both categories actually. And you can see hydroxychloroquine is in there as well. Losartan's being looked at. Some people thought it may worsen conditions. Some people think it may actually improve. So that's being looked at, which is a blood pressure medicine and also used for people that have kidney issues in general. And so there's a big list here, some of which are HIV, antivirals. And then there's under treatment candidates expected to enter clinical trials by the fall of 2020, there's actually 15. And human antibodies is one of these categories, which is what we were just talking about. There's really no early news that I know about yet regarding any of these treatments. There's been a few, for Plaquenil, which is the hydroxychloroquine, there's at least three studies going on, or that have taken place, or it might be 4 now, and 2 showed effects and 2 showed that it didn't work. So I think it's up in the air, and now there's larger studies that are randomized controlled clinical trials that are properly being, at a high rigor, scientific rigor, that are now happening in real time as we speak. And we've talked about those before, so we should learn about those pretty soon. There's these studies that you could consider platform studies where, and you can see there's 4 large ones, the WHO Solidarity Study, where they're looking at multiple different treatments. So you get randomized to one of 4 different arms. And you can see here the different sort of treatments they're doing. Interferon is the IF beta 1A. And so that's one example. National Institute of Infectious Disease has this adaptive study. UCSF and others are part of this. This is a global study. I believe Singapore is part of that, and a couple other countries, as well as multiple sites in the US. And I believe their study is full, or I think they enrolled everyone already with remdesivir, so we should hear the results soon. And then they quickly are going to do dose changes or add other drugs based on the results that they find. So that's why, hence the adaptive term in the study name. And then the recovery trial, which is the randomized evaluation with COVID therapy. Again, multiple different arms. This has the hydroxychloroquine, which is the Plaquenil, with the Z-Pak, which was the original, originally looked at in the French study that showed a positive result and got all the press, which made President Trump announce it to everybody. And then you've got the steroid treatment too, which is the low-dose dexamethasone, and then the HIV drugs. And then you have the RMAPS and CAP study. And again, they have multiple arms here as well. So we'll get data soon. The good news about COVID-19 is it's a quick turnaround. It's not like something that you live with and the outcomes are a year down the road. The outcomes are pretty immediate. So therefore, and the good and obviously bad news is that many people are infected and many people are in the hospital. So we can quickly get data. Enrolling people is not a problem. The length of illness is short. So we should get high turnover, so leading to results relatively quickly. There's a lot of consortiums being formed, which is just amazing to me and beautiful to see. There's so much competition in academics and in the private sector with biotech and pharmaceutical drug companies. As I mentioned, I think it was last week about, the masks that are being made that are going to every home in America, right? 125 million homes. So 600 million masks are being made. And that was done by all these textile groups that came together that are normally in competition, and they created a consortium. And again, that's happening here. So here's an example where there's a partnership brings together the world's leading plasma companies to focus on this. And you have these companies here that created this consortium and are really doing this work. Now, the plasma, and we spoke to this a little bit last week, relates to the fact that you have someone who's sick, they had therefore developed antibodies. And then what you do is harness those antibodies because they're in the plasma. And then what Bernadita was saying, Dr. Mendez, was then you give that then to a sick patient who doesn't have the antibodies yet. And hopefully they do a lot better. And so there's studies being done looking at that as we speak. And at the same time, there's the hope that we can develop these monoclonal antibodies where you could basically take those antibodies and then scale it. And I'd love to hear, Dr. Mendez, because you know about this work, you guys have done this work. How does it work that you develop these? And Dr. Gutierrez, if you know about it as well, I'd love to hear, how do you take these and make these monoclonal antibodies where it's scalable, where you don't need to keep drawing people's plasma, and you can create a platform that does that in the lab?
Okay, I before that, I wanted to make one comment. It's interesting that if you look, you know, the first table that you put when you were detecting antibodies at different times,
Yeah.
the antibodies really started to pick up like at day 15, isn't it?
Yeah, I'll show you right here.
Right. Look. So you see that at day 15. So really what happens is that people who are really in bad shape are fighting all that time without any antibodies. I mean, any IgM or IgG much later, but any IgM at all. So obviously, the idea that you can give these people, you know, antibodies that come from somebody else, it's very attractive because it can certainly help in the fight against the virus if you find neutralizing antibodies. And that's, you know, we go back to the previous conversation. Now, monoclonal antibodies, there are different ways in which you can do monoclonal antibodies. I mean, you have to generate cells in the labs, they do them in bioreactors, but there's some interesting production methods now in animals. You know, llamas, they're starting to use llamas and goats and things like that to do them. So I wouldn't be surprised if we see both, you know, some companies working in the bioreactor mode and probably going to something that can give higher scales faster, because the bioreactors are, you know, the scaling up is always an issue. So that would be my comment there.
And the polyclonal hyperimmune immunoglobulin medicine, was that, what does that fall into when they speak about that?
I don't know that much about that, really. So I don't know if,
Mary,
I don't know. I never worked with it, really.
In terms of using a polyclonal antibody, Brad, versus a monoclonal, like the plasma. I haven't, I know that we have used, I sort of mentioned it before, we have used a, you know, a combination of immunoglobulin against a virus to use to establish protection against another virus, in the case that I used earlier. But I think really the route to go is via the monoclonal antibody pathway, and maybe a mixture of monoclonal antibodies that can elicit protective efficacy. There's great genetic engineering technology out there. I know they're already, if they're looking at neutralizing antibody, they're going to be mapping it out so they know exactly what proteins on the virus need to be bound to antibody in order to prevent the virus from infecting a cell. So when we talk about neutralization, we talk about protection, we are talking about that actual, you know, physical biochemical response. And they're able to determine that. They're doing sequencing, I'm sure, on if they have identified the neutralizing antibodies and the proteins on the virus. They're going to be mapping that out carefully, and they can begin engineering antibodies based upon the information that they find. Again, that takes time, but I know there are groups that are probably very excited to take that challenge on.
There are actually some companies that already have started working on it because they had a head start with some previous work done in other coronaviruses. So I'm sure that we'll be seeing some of that also soon. The use of antibodies, you know, from plasma is something that can be done, but very, you know, on a very small scale, because, you know, probably from plasma of one patient, you can treat, I don't know, in the order of 10 people. I don't know, but it's just small numbers. So really, there's no way that you can generate the production level that you need doing that. That's kind of an emergency initial situation, and it's kind of a validation. It's a proof of concept situation.
Yeah, my understanding is the hope is, you know, by the fall that we might have a monoclonal antibody. That's what's on the biotech side is what I'm understanding. Yeah. The nice thing about this particular consortium is they're trying to develop, one unbranded anti-SARS-CoV-2 polyclonal hyperimmunoglobulin. So, they're really unbranded, all working together. It's a beautiful thing to sort of bring everyone together for efficiency.
And FDA also allowed this to be a treatment, opened it up basically for the sites.
Yeah.
Yeah. And I think, Brad, this whole discussion helps inform a potential response to Ken's question. I don't know if you saw that. For those who are over 60, we're talking about the long haul. If we're talking about antivirals, such as or treatments, such as this antibody potential, how that will impact people's ability to move about in the coming year.
Yeah. So I was going to move to the next slide, which talks about our sanity.
There you go.
Are relevant to that question. But I think let's talk about it from a practical level first. We're in a hard spot because to reintegrate now when we're all naive to this virus, there's risk. So the only way to do that is to really start to test the population to know, how many people are showing up in the hospital. Okay, as that really comes down, which by the way, I'll just tell you, I was at the Veterans Hospital today. I've been involved with a lot of the UCSF calls that have been going on weekly and learning about the census, as well as the San Francisco Department of Public Health and others where we know about the census for all the hospitals in the Bay Area. And it's really low. We prepared for disaster, and thankfully we haven't had to act on that. So for San Francisco, at least, we're all in different places, but for San Francisco, at least, if we start to track and be able to test and see that the cases are going down, only then can we start to reintegrate, right? But we don't even know the immunity level in the population. So there's a lot of unknowns. So there's a lot of risk. So I don't see this happening at a large scale for a while. I think it's going to have to be done very methodically, where people, have to change their workplace orientation, or they're going to continue to have to limit grocery store, how many people can be in the grocery store at a certain amount of time. What's going to happen in movie theaters? What's going to happen, right? All the places where there's crowds, how are you going to occupy a stadium for a sports event? Will it look like the U.S., the White House press briefings where everyone's sitting, every fourth chair or whatever that might be? So I think that's what's going to have to be in play. Clearly, people are going to be wearing masks, so I think that's a big part of the equation here. Will be, masks and gloves and precautionary measures throughout. It's a different world order, for sure. I don't know if either of you have a comment.
It was interesting to me, Brad, the other day. I was very impressed when all the news came out on Monday about the governors getting together, the governors in the Northeast, and of course the governors out on the West Coast. And in California, a very logical list of criteria that would have to be met in order to open up. And one of those criteria, I think it was the last one on the list, is the redesigning of offices and other places of gathering. And I felt that was fantastic to see that that's the type of forward thinking that's going to have to come into play for this. And we just all have to accept that as a new reality, and everybody's going to have to wrap their mind around it in terms of how we conduct our business. So, but at least there's some very proactive and what I would consider positive dialogue out there regarding it. Whoops, we don't hear you.
So one of my friends is a restaurant owner. He owns several restaurants in Marin. And his big question is, okay, so I can bring my I can test all my employees. What about all the customers coming into the restaurant? How's that put us at risk, and how do we redesign the restaurant. So again, I think these are all relevant conversations to have. Are people, are the waiters going to be wearing gloves? Or are they going to be wearing masks? You know, what does this look like? I think it will be very different. Bernie, did you have any other comments?
Not really.
Okay, so let me just spend a moment then. Thank you both. I wanted to talk from the mental health part of the equation. This was just released today, Lancet Psychiatry, you know, talking about a call for action for mental health. And that's pretty major. It's quite timely and it's very true. And it leads to the question that's being asked. That really sort of bodes this question. And what one particular, you know, part of this, I think it's relevant. You know, an immediate priority is collecting high quality data on the mental health effects across the whole population and vulnerable groups on brain function, cognition, mental health. There's an urgent need for research to address how mental health consequences for vulnerable populations can be mitigated on the impact of repeated media consumption and health messaging around COVID-19. And I think that last phrase is the most relevant. And this is in part why I'm hosting these webinars. Is my intention is to give you a place to learn and ask questions so you don't have to focus on the repeated media consumption and health messaging. Maybe the health messaging that's relevant for public health, you know, but all the media, I mean, that is all, if you pick up any paper, or I pick up the paper, but a lot of people that read it off their tablets or whatever, or you know, the news or whatever, it's just overwhelming. And it gets to a point where it really beats us down and can be emotionally very trying. So with that in mind, I think it's really important to find a place to ground ourselves in to find our own sanity, you know, and to intentionally limit the amount of information that's coming into our purview. I had to do that initially when there's a change of office at the White House. It's to sort of limit the amount of exposure. So my own sanity, everyone has to, but I think thankfully this is, you know, crosses all political boundaries, and this is relevant for all of us. And it's real important to sort of, at this point, there's less new information happening from day to day, from week to week. It's, you know, and so I'm hopeful that by doing this, whether I do this every other week or we do this weekly, whatever the timing feels right for all of us, that we do it in a way that really allows for us to come here and have a conversation, and then limit the amount of input that's coming from outside, because there's so much happening there. So I hope that answers your question. And you know, we've come up upon the hour, and I want to be true to our time. So thank you all for participating. Dr. Mendez, Dr. Gutierrez, always value your comments.
Thank you.
Hopefully we've got a nice summary here of where we can actually sort of understand a bit of the statistics that's happening. And we talked a bit more about the diagnostics, which I think is quite important for us to understand. As we learn more about treatments, I'll be sure to include that when there's specific, you know, outcomes that are of note that I think are quite important for us to keep in mind. And I thank everyone for their questions, and I look forward to seeing you next time. So until then.
Thank you.
Stay healthy.
Thank you. Take care, everyone.
Yeah.
Bye-bye.
Bye-bye.